Hello, my name is Ana Veloso, and today I'll give you an introduction into the SynEcoSys single-cell RNA-seq database. I'll start by introducing you to this database, then I'll demonstrate its main functions, and finally I'll guide you to a case study to illustrate how it can be applied to research. But let's start by the importance of single-cell sequencing. And compare with bulk sequencing is the same thing as comparing a smoothie to a fruit bowl. So even though they have the same components, the readout that you have from it is quite different. While from bulk sequencing, you only get the average expression profile of the entire sample. From single cell sequencing, you are able to take the pieces apart and identify the individual cell types and the differences between the cell types and how they communicate with each other. This can be applied to a multitude of fields, but is particularly interesting in where there is a high heterogeneity, such as cancer. Well, you not only want to understand the cells that make up the tumor, but also the communication with their environment. In neuroscience, this can help you classify neurons and how they interact with each other. And immunology, it's important to be able to identify all of the immune cell subtypes and to understand the mechanisms that modulate the immune system response to a particular disease. Finally, in developmental biology, you can dissect the gene expression patterns that occur during developmental processes. So this field has been growing quite fast in the last few years, and a way to look at this is to look at a number of publications that come out every year that use single cell. And you can see that just last year, over 2,500 papers were published. And with this comes positive things. So we have massive data sets that cover most tissues, either in healthy or diseased states. And exploring these public data sets can save you time and cost in the lab and can complement your own data and allow you to draw more robust conclusions. But there's also some problems. There's a high variability in data analysis. Data analysis requires both experienced bioinformaticians and a lot of computational power. And there's also some research biases, so you cannot directly compare two data sets that have been published. And to solve this, we have SynEcoSys, which is a standardized data collection platform with clinically relevant information and easy to use data visualization tools. So it's not just a repository of data. In SynEcoSys, we collect standardized public data sets, we annotate each and every sample with every clinical and prognostic information that's available. We go through the paper to the supplementary information, and all information that we can find is added. Data is run through a standardized pipeline, CellLens. The annotations are manually curated, and this allows for direct comparisons between every data set present. We also have database-wide statistics of gene and cell type distributions. And finally, the visualization tool, CellVis, allows us to visualize the data in various different ways, which I'll go into more detail later. So we have a lot of experience with cell annotation. We have sequenced millions of single cells, and we are able to automatically annotate over 2,000 different cell types in human, mouse, and non-human primates, which is the data sets that we have in SynEcoSys. Our annotation is quite accurate. We usually either have a one-to-one match with the original published work, or we are able to identify subtypes of a larger cell population. It's also important to note that we unify the terminologies of cell types, so it's easy for you to compare different data sets. It's not only about public data, you can also use it to analyze your own data. And for this, you can upload your expression matrix, run it to the same automated pipeline, which will annotate your cell types. This annotation is not final. You can manually change it and curate it. And you can then perform cell visualization analysis by using CellVis. A very important point for SynEcoSys is clinical translation. As I mentioned before, we make sure that we had molecular level patient stratification. We had every clinical and prognostical information available for the samples. And in here you can see a snapshot of some of the data available for one of our lung cancer data sets. We have information such as cigarette use, mutation detail, differentiation grade, response to treatment, stage of disease, or other concomitant diseases. And this allows you to then just click on these boxes and compare, for example, gene expression between a patient that is resistant versus a patient that is responsive, and try to understand what are the mechanisms behind this resistance to treatment, for example. And this is all wrapped in a user-friendly interface, as you can see here. Now I'd like to guide you to the various ways in which you can explore the dataset. So first, in the home page, we have data statistics, how many data sets, how many cell types, how many marker genes, and this you can also see plots. And here I'm just showing you the data sets per disease subtype. Currently, we have over 200. This number is rapidly changing as the data set is updated every week. Around 10 publications are added. And you can search the database by genes. Just type the gene of interest, select the correct species, and then perform a quick search. In here, you'll have the gene basic information, such as gene name, species, protein function. We also have the pathways that genes involved in by the reactome and the wiki pathway, and also drug targets. So if a drug has been developed that targets a specific gene, will be listed here. There's also bulk RNA-seq information, where you can see in which kind of diseases or tissues this gene has been found previously. Within SynEcoSys, you can see what are the top 20 cell types where this gene has been found expressed. And for each one of these cell types, we have in which tissues were these cell types found, as well as if these tissues were from healthy or disease samples, as you can see here. Underneath, you'll be able to see the core dataset icons, and this is just data integration of multiple publications for specific diseases. Underneath, all the publications that have the expression of the gene you search are here. If you click on View, you have a short summary of the paper, and the next tab, you can do the data analysis and visualization. Now, if you go back to the home page, you can also search by cell types. So just type the cells of interest, select from the drop-down menu, and this will give you basic information about the cell type and also canonical markers, not just for human, but also for mouse. We also have cell ontology, so you can just click on the plus and minus so you can see the progenitors and the precursors of a specific cell type. Underneath, we have the top 20 tissues where these cell types are found in our database, and you can also see if they are from healthy or disease samples. Again, underneath, you can again find the core data sets and the studies. Going back to the home page, another way to explore SynEcoSys is to look at the cell atlas. So you can move through human, mouse, and non-human primates. Then you can select a system. In that system, you can either select tissue, and from that tissue, there's different subparts. If you click on one, for example, cerebellum here, it will take you to all of the publications that have data related to the cerebellum. If you go back to the tissue atlas, and now instead of selecting tissue, select a cell type, and click on a cell type, you'll have the canonical markers for the cell types. This list of markers is an in-house list and is the one used for the automatic cell annotation. Finally, you can browse the dataset by study, and you can use words in the title, the SynEcoSys ID, or the DOI. And you have also stated statistics about the diseases that are mostly represented in our database, as well as information about the sample types. For example, you can see here that most of our samples come from tissues. Again, you have the core data sets and the publications. We also have resources. There's a documentation telling you which software you use for the analysis and how it's done. The guidebook is a step-by-step description of how to use SynEcoSys, frequently asked questions, and contact information. In the data analysis portal, you can look through the analysis that you've done previously. And here I'm just showing you how you can go back to a trajectory analysis that I've done by clicking on View Results. The settings that you use for analysis are saved, so you can just adjust them to your needs. You can initiate a new analysis by importing a data matrix. For the annotation, you get to select which software you want to use, as well as some more settings, and you also can import your metadata by using our template. And finally, you can go to your account for some basic account information. So this was just very quick, very summarized description of how you can explore the database, but I think most importantly, it allows to apply it to scientific questions. And SynEcoSys can be used to explore mechanisms of a disease or of a cell interaction, how to find potential drug targets, to verify your predicted drug targets into larger data sets. And today I'm going to show you an example of this. So I think this is very focused on the power data mining. So this Nature Communication publication shown here, the authors performed single-cell RNA-seq analysis in samples of 6 patients of triple-negative receptor breast cancer. Another group decided that it would be interesting to explore this data to try to find targets for immunotherapy, and they did this, and they found that the interaction with CD24 cyclic 10 seemed to be cancer-specific. And now I want to show you how easy it is to verify this in SynEcoSys. So again, this data is available, so you can just easily go to Browse by Study. You can type some keywords, in this case just TNBC. You can then navigate to the correct publication and click on View. This will send you to the Paper Information tab, and you can then go to Data Analysis. Because we want to verify this gene expression of this interaction that the authors found, you can just move down to gene expression and type the genes of interest. In this case, CD24 and cyclic 10. And when you click search, feature plots will be generated where you can see the expression of different genes. You can see that CD24 is expressed in the cancer cell and cyclic 10 is expressed in the macrophages. You can also see in the column plot here that CD24 expression is restricted to the cancer cell. So this confirms what the authors also saw, that CD24 is exclusively expressed in the cancer cell and cyclic 10 in the macrophages. And the authors tested if using an antibody against CD24 would inhibit this interaction, and it did. It did. And when they inhibit the interaction, the macrophages were able to perform tumor clearance by phagocytosis. And it did a lot of validation experiments. So this interaction is responsible for inhibiting tumor-associated phagocytosis. But the data that they explore only contains one subtype of breast cancer and data from only 6 patients. And as we know, cancers are all highly heterogeneous, so it's important to explore larger data sets. We have one of these present in the SynEcoSys. So this publication from the EMBO journal has data from 4 breast cancer subtypes, including the TNBC, and gathers data from 69 patients. So now I will show you how you can verify if this interaction is still specific in this data set. So again, you go to Browse by, click Study, and type some keywords. You can then again navigate to the correct publication and click on View. When you go to Data Analysis, you can color the UMAP by disease subtype, and you can see that there's 4 different kinds of breast cancer: estrogen receptor positive, HER2 receptor positive, progesterone receptor positive, and triple receptor negative breast cancer, as long as healthy tissue, which is labeled here as normal. If you scroll down, You can again type the genes of interest, CD24 and cyclic 10, and the future plots will be generated. If you arrow with the mouse on top of the UMAP, you can see in which clusters the genes express. And you can see that it's expressed in the epithelial cells as long as in the cancer cells. So the expression of CD24 is not exclusive to cancer cells in this case, while the expression of cyclic 10 is still exclusive to the macrophages. We can further compare this by selecting cancer cells in epithelial cells in healthy tissue versus the cancer cells and epithelial cells in breast samples, cancer cells. And this will generate heat map. In this heat map, you can see that the levels of CD24 are the highest in breast cancer cells, so cancer cells from breast cancer samples, but they're also expressed in normal tissue, in epithelial cells, and epithelial cells of cancer in cancer samples. So expression of CD24 is not exclusive to cancer cells, which affects their value as immune therapy. Of course, the differences, and we see that these differences are probably due to patient factor. It could be age or some another mutation that we don't know about that needs to be further explored, and this could also be done in SynEcoSys. But the fact stands that verifying your targets in silico can save you time and costs by guaranteeing that you only choose the more robust targets for your further experiments. I want to mention that the visualization module of SynEcoSys is very complete, and today I focus only on one of the modules, the gene expression, but there are 4 more. You can explore cellular composition, compare the numbers of the ratios of the different cells from different patients. You can look at differential expression using any of the factors in the metadata and clinical information. You can look at cell interactions, and this looks at receptor ligand or receptor interaction of different cells. You can see here one plot representing this, and you can also perform trajectory analysis. So SynEcoSys is the single cell sequencing data platform to use for clinical translation, as we have very detailed annotation with clinical and prognostical information, and it's easy to validate your results and find new insights with a few clicks, as I showed you today. So I'd like to thank you for taking the time to watch this video, and if you have any questions, please don't hesitate in writing us, and you can find more information in the website and on our LinkedIn page.